Okay, so I want to go right into introducing our first speaker, Patricia Wittkopp from University of Michigan. She's done many, many wonderful things in the area of transcription, evolution, using both flies and yeast, and I'll just keep it short and give her her full slate of time.
Thanks, Mike. So you stole my two things I was going to say. Number one, thank you to the efforts of you and the rest of the organizing committee for putting this together. This is, looking at the schedule, fantastic few days ahead. So I'm really excited and honored to be a part of it. Throughout my career, my research has been motivated by trying to understand how phenotypic differences arise. And we've learned in the last 20, 30 years or more that changes in gene expression are a really critical piece of how that happens. This suggests that if we can understand how gene expression evolves, we're moving closer to understanding how higher order phenotypes evolve. And so much of my work in the last 10 to 15 years has focused on that question, trying to sort of treating gene expression itself as the phenotype and trying to understand how it evolves, specifically transcriptional regulation, which is the first step in gene expression. So transcription, you said a right click, right, or a left? Left. I'll go with the old-fashioned way, hopefully. There we are. Okay. Transcription is sort of at a very basic primary level controlled by interactions between cis-regulatory sequences and trans-acting factors. Changes in either of these components can give rise to differences in gene expression. Again, we've been studying the relative contributions of changes in cis and in trans to expression differences both within and between species. I'm just going to share with you one sort of key observation from that work today to motivate the rest of the talk. That observation, we have a lag or something here. That observation is that the contribution of cis and trans regulatory changes to differences in gene expression or transcript abundance change over evolutionary time. So they evolve. And cis and trans are not contributing equally to the evolution of gene expression. So as I'm illustrating here with some data from yeast, so we have a comparison of two strains of Saccharomyces cerevisiae. Now it's going to jump ahead without me, huh? Okay. As well as other species of Saccharomyces. And we've compared gene expression across the genome between these strains and species and measured how much of those expression differences are due to cis or trans regulatory changes. We boil those measurements into something here that I'm calling proportion cis, which just asks, okay, of the total amount of regulatory divergence that's occurred for a given gene, how much of that is attributable to changes in cis regulation? And the interesting and unexpected observation is shown in this box plot here, which in each case summarizes that proportion cis for all of the genes in the genome in a comparison, again, of the two strains or the two different species with increasing divergence time. And what we see is that the amount of contribution that cis regulatory differences make is increasing preferentially, faster than trans regulatory differences over evolutionary time. So we observed this pattern. Again, it was unexpected, and so we're kind of trying to explain why. Why do we see this? It's now been seen in flies and yeast and hints of it in mice. And this is a point where I realized that if we're going to make sense of the patterns of polymorphism and divergence that we see in natural populations, I think we also need to know more about the raw material that gives rise to those, specifically the effects of new mutations. So new mutations produce variation in a trait, including gene expression. Some subset of those mutations survive to be sampled as polymorphisms within a population, and some subset of those give rise to fixed differences between species. So the goal of the work I'm going to tell you about today is to better understand the mutational input for regulatory variation and how it sort of is filtered through time, through the action of selection and drift. Okay, so what I am going to do today is give you highlights from some published work and then also share some unpublished data with you. First, I'm going to tell you about work we've been doing comparing the effects and frequency of cis and trans-acting mutations affecting expression of a single gene. Okay, so just how do these two classes differ? I will then tell you how we've used that baseline data of mutational effects to compare to the effects of naturally occurring polymorphisms to infer the role of selection. And then I'll end with the unpublished data where we're using fitness assays in the lab to better understand how changes in cis-regulation affect fitness. Now, the work that I'll be showing you today was done primarily by these 5 people, so I want to take a moment to highlight them. First is a former postdoc in the lab, Jonathan Gruber. Next are 2 former graduate students, Ryan Metzger and Dave Yuan, and 2 current postdocs, Fabian DeVoe and Andrea Hodgins-Davis. Okay, so before I get into what we found, let me tell you a little bit about our study system. The system we're using uses a promoter from a native yeast gene, a gene called TDH3, and uses it to control expression of a yellow fluorescent protein. We put this protein into haploid yeast cells as a way to easily quantify expression of that gene. TDH3 is a gene that encodes an enzyme that functions in the glycolysis pathway. It does have fitness consequences if deleted, but it's not lethal. There's a paralog TDH2 that has some partially overlapping function. It is highly and constitutively expressed throughout the cell cycle, so it's very abundant and easy to study. I wish there was a better way to advance slides. I don't know why the keys aren't working.
There you go.
Just okay. All right. Thanks. Okay, so it's highly and constitutively expressed. The promoter sequence controlling its expression is largely conserved among other Saccharomyces species. That's shown at the bottom down here. Its promoter has been characterized a bit. We know specifically that 2 different transcription factors, RAP1 and GCR1, bind to these binding sites and are critical for the activation of this gene. And finally, an advantage to working with yeast in general is we have these large functional genomic data sets that allow us to infer at least potential regulatory connections of who might be regulating who's, ChIP-seq type data sets. And from that, we can think about how variation might be structured within this regulatory network. All right. Okay, so if we're going to study cis and trans-regulatory mutants, we need cis and trans-regulatory mutants. Our prior work showed that if you just try to introduce mutations randomly, cis mutations are very rare relative to trans mutations. We'll come back to that point. So we introduced our cis mutations systematically. We marched across this promoter, changing virtually every G to an A and C to a T. For the trans-acting mutations, we introduced those randomly with a very low dose of EMS. So EMS is a chemical mutagen. It also changes Gs to As and Cs to Ts. We used a level of EMS such that only 1 mutation that alters expression of the reporter gene is induced per cell, although there actually are about 30 base changes in the cell. Okay, Gs and As and Cs to Ts are obviously a subset of the possible point mutation types. However, they are the most common type of spontaneous mutation in yeast and also the most abundant polymorphism. So if you have to pick a subset, it's a pretty good subset to pick. All right, so we have these yeast cells expressing this fluorescent protein. Each one has a mutation somewhere in its promoter or its genome. We grow up 6 replicate liquid populations of each genotype. Each of those is analyzed with flow cytometry, where we get measurements of fluorescence for each individual cell. Those estimates allow us to calculate a mean expression level for that one replicate of the genotype, as well as a variance, or the standard deviation, a property that's related to noise, the consistency of expression among genetically identical cells. Okay, so what have we found? Again, first I'm going to give you some highlights of published work comparing the frequency and effects of cis and trans-regulatory mutations, because they don't provide an equal source of variation, even for this one gene's expression. So shown here are the effects of those individual point mutations that we made. The largest effect mutations, all of which decreased expression, are in the previously identified binding sites. So that's interesting. Most of the other mutations we made have smaller, well, they all have smaller effects than those. We don't see other obvious clusters of large effects suggesting a lot of other binding sites at least. Okay, what we're more interested though is what does the distribution of those effects look like when you put them all together? And so I'm just representing that same data here differently. Colored in black are the mutations in those transcription factor binding sites. Okay, so this is the distribution of effects of cis-regulatory mutations in this gene. We can compare that then to the distribution of effects of the trans-regulatory mutations. And when we do this, we find, number one, that we don't see large effect decreasing expression mutants like we get with the cis-regulatory mutation set. We also can see that their average effect size is smaller. And to illustrate that, if we basically fold these distributions in half and ignore the sign, we can get these box plots here that clearly show that cis-regulatory mutations tend to have larger effects on this gene's expression than trans-acting mutations. Okay. So our other question about the relative frequency is actually a little trickier to answer because it depends on how you define a cis or a trans-regulatory mutant. So stepping back, the potential target size for trans-regulatory mutations is obviously much bigger than for cis-regulatory mutations. It's essentially the whole genome, 12 megabases in yeast. The cis-regulatory mutation target size, we think, is about 678 base pairs, which is the size of the promoter. If we consider, however, the proportion of mutations in each of those two groups, or so the absolute number of mutations that have effects of a certain size, we can clearly see differences in the relative frequency that are not always intuitive. So what you're seeing here is a summary. So if we look at 100%, this would be all mutations that would keep expression the same or change it at any level. So it's sort of that potential space. So in red, we have cis-regulatory mutations. In blue are the trans-regulatory mutations. And this question about what's more common, cis or trans-regulatory mutations, actually depends on what cutoff you use. For instance, do you need to change expression in a statistically significant way or a biologically meaningful way, something that affects fitness? Or do you want to put a hard percentile sort of cutoff on that? You can sort of, this is just showing sort of the full distribution. And so the main thing I want you to get from this is that cis and trans-acting mutations are not generating identical distributions of variation for natural selection to act on. So for instance, if you ask about mutations that decrease expression at least 10%, cis are more common. We've yet to recover a trans-acting mutation. If you want mutations that increase expression, trans is actually more common. Okay? The brown points I haven't told you about. This is where we repeated the EMS experiment but selected the most large effect, right? So the two tails to try and get extreme effects we might have been missing because of their rarity in the trans collection. It doesn't change the distribution much. Okay. Another way that we found cis and trans-acting mutations to have different effects is in their dominance. So we isolated all these mutations in haploid cells because we didn't want to miss recessive mutations. And then we tested their effects in diploid cells. So if we compare the effect in haploid and diploid cells and find that a mutation has the same effect, we would consider that additive or dominant, not recessive. If, however, a mutation has an effect on a haploid cell but not in a diploid cell, we would classify that as recessive. Okay. When we look at the effects of cis-regulatory mutations, this is obviously a small subset. It was actually done in earlier work. We see that they have similar effects in haploid and diploid cells. Trans-regulatory mutations, though, tend not to. So trans-regulatory mutations tend to be recessive much more often than cis-regulatory mutations. And the level of dominance can affect sort of the fate of a mutation in a population. So the patterns of polymorphism and divergence that I told you we observed in yeast, and we've seen in flies, and other groups have as well, where there seems to be a lot of trans-regulatory variation within a species, but when we look over increasing divergence times, cis mutations seem to accumulate preferentially. And we think that these mutational properties might be part of the explanation for that pattern. So for instance, trans-regulatory mutations arise more frequently, but cis-regulatory mutations tend to have a larger effect when they do arise. They also tend to be dominant, and so selection can see them more easily and act on them. Pleiotropy has a question mark next to it only because I think that we're still missing some really great empirical data supporting this, but the common wisdom is that trans-regulatory mutations are more likely to be pleiotropic and therefore more likely to be deleterious when they arise. Okay, and that's also consistent. Okay, so as I said, our main motivation for collecting these distributions of mutational effects was to have a baseline to compare to variation that exists in natural populations. And so we did just that for the cis-regulatory variation specifically. So the idea behind this test is that if you know the distribution of effects from random mutations and you compare that to the distribution of effects of polymorphisms you actually find in the wild, if those two distributions are equivalent, we can't invoke selection. Right? This could just be the random effects of mutation and drift. If, however, we see a difference between these distributions, we can, number one, infer that selection acted, and number two, say something about how it acted. So in this cartoon example, we might infer that selection had favored mutations that increased the trait value or disfavored those that decreased it. Okay, so if we're going to compare our mutations to natural variation, we need natural variation. We've taken advantage of a collection of yeast strains that have been isolated from a variety of environments. There is variation for the TDH3 gene in those. Some of that is caused by cis-regulatory variation, and of that cis-regulatory variation, 97% of it is explained by variation in the promoter. So we're going to characterize that promoter variation. When we look at the sequence of this promoter in these natural isolates, again, we see that there are polymorphisms, but the frequency of polymorphisms in the promoter compared to the frequency of synonymous changes in the coding sequence of this gene is lower, suggesting that purifying or negative selection has acted on this promoter, okay? All right. Despite that, when we compare the distribution of effects of these mutations and polymorphisms on mean expression level of this gene, we see no statistically significant difference between these two distributions, which says that we have no evidence that selection is acting on mean expression level here. Okay? I want to remind you that actually very few mutations changed mean expression level in a large way. So I think it has to do with the rarity, not that expression level of this gene isn't important. When we make the same comparison, however, looking at noise, which again is the variability among genetically identical cells, we do see a difference between the distributions of effects of mutations and polymorphisms. Specifically, we find that the polymorphisms in the wild tend to maintain a lower noise than a random subset of new mutations would. Okay, so we find evidence that selection is preferring variants with, or polymorphisms, genotypes, with lower noise in natural populations. Now, if you don't think about noise in the context of fitness a lot, and I certainly didn't before we observed this result, I think this is one way to think about it here. So we have two different genotypes shown in red and blue, both of which have the same average expression level, but they differ in their variance or their noise. If that average expression level is the optimal state, then the genotype with the greater noise, blue, is going to produce more individual cells that have abnormally high or abnormally low expression, making them less likely to divide. Okay, so when averaged across all the cells with those two genotypes, the red we would expect to be more fit in this case. Okay. So we see a stronger signal of selection acting on expression noise for the TDH3 promoter than on mean expression level. I want to be clear, I don't think that this is because selection for noise is stronger than selection for mean expression level, but rather that new mutations are more often changing noise than they are having big changes in gene expression level. So I think the signal of selection we see as a product of the mutational bias that is just the reality of the system. Okay. So one of the things that we're trying to do now is better understand how selection may act on individual cis-regulatory variants, specifically with their effects on mean and noise. So this is the unpublished work that I mentioned that's primarily Fabien Devaux's work here. So what we did was to take our collection of mutants, cis-regulatory mutants, where we had, or a subset of them, where we had initially introduced the mutation in front of YFP and reintroduced those into a genetic background that has a sort of a common reference YFP, put them in the native TDH3 promoter. So now we're looking at their effects when we change the expression of native TDH3. We could then do competitive growth assays starting with two labeled strains and measuring sort of how they grow through time as a proxy for fitness. In yeast, that's basically fitness. Okay, so in the first pass of this experiment, we selected genotypes that altered expression to the levels shown here with arrows. So we had a wild type level, one that increased a little bit, decreased a little bit. Some of these big decreases were, again, those mutations in transcription factor binding sites. And we also looked at a complete knockout, so what happens when you lose this gene's activity altogether. Shown here is the fitness curve for that, so what are the relative effects on fitness for each of these cases. And what we see is that if you decrease expression, even up to almost 10%, we have a no statistically significant, detectable in the lab change in fitness. Okay, and this assay is quite sensitive. Those are 95% confidence intervals, and we can detect a 0.3% change in fitness. So compared to the effects of new mutations, what we find is that most new mutations are unlikely, the effects of new mutations on mean expression level are unlikely to be affecting fitness rather, are unlikely to be affecting fitness, and that's consistent with our previous data. Now, we wanted to better understand the signal that we saw in the other data with noise, and so we created some additional genotypes that will sample both increases in expression level better, decreases sort of more fully, and then also we were trying to sort of decouple the correlation that exists between mean and noise. So we did this by creating various combinations of mutations. We also started mutating the Tata box, which wasn't mutated initially because there wasn't a G or a C in it. We also made some lines that had 2 or 3 copies, sometimes with mutations in those, basically just trying to spread out and sample a range of mean expression levels and noise values. So these are the points, the genotypes we analyzed. So this is the range of mean expression level, going from, again, complete loss of 0% up to 300% of the wild type level. And you can see that changes in mean expression are correlated with effects on noise, but they're not perfectly correlated. And so what we want to know about is if we have genotypes that have similar mean expression level but different levels of noise, how do they differ in fitness? Okay? And so to do that, we've been, this is sort of a first pass analysis. Not the results I'm showing you are not preliminary, I fully believe them, but I think there may be more sophisticated ways to do this. So what we're looking at this stage is sort of the differential between the amount of noise observed and what you would expect based on sort of this full set projection, I guess, of the effect on mean expression level. And so we can compare that differential for noise, which is uncorrelated with mean, to the differential in fitness effects. Okay, so if we change mean expression a certain level, there's a certain change in fitness we expect. And what we're looking at now, again, are the points that deviate from that expectation. And then what I'm going to show you in the next slide is a comparison between the deviation in noise and the deviation in fitness. Okay? And I'm going to show it to you in 2 different groups, and then I'll explain why. So the first group here are the genotypes that decreased fitness at least 1%, which happen to also all decrease expression by about 70% or to 70% or less. What we see in this case is a statistically significant correlation that says that greater noise actually equates to or translates to greater levels of fitness. That's the opposite of what I told you already, so if you're watching closely. Okay? If we look, however, at, let me back up for just a second. If we look at these points, which have higher fitness, but again cover a wide range of expression levels, we see actually the statistically significant opposite direction of correlation. Okay. So what we think is going on here is that if you have a genotype that produces mean expression level that's close to optimal, noise is bad. You produce more cells that are far away from that optimum. If, however, you have a genotype whose mean expression level is suboptimal, noise is good. You produce more cells that have phenotypes, more individual cells that have higher fitness phenotypes. And there's some work, and we're doing some more modeling work now to show sort of the rate at which those can actually have a big advantage for the population, bigger than this plot would suggest. Okay. So the take home from all of this sort of data together, especially the last part, is that changing levels of gene expression, number one, has a sort of nonlinear effect on fitness. Others have seen that for other traits, that's not surprising. And then that the effect of noise on fitness, I think, is really going to depend on where you are, where that genotype is relative to the optimum. All right, so I've tried to thank people along the way, but again, to reiterate, all the work I showed you was done by a very talented group of researchers, Brian Metzger, Fabian Deveau, Dave Yuan, Andrew Hodges-Davis, Jonathan Gruber, Bing Yang's work I didn't talk about today, but he's also doing some interesting stuff on compensatory evolution with help from this group down here, and the yeast data I showed you was Joe Kula and Brian Metzger as well, and others in the group, including Joel McManus as a collaborator who's in the audience today. So thank you, and I'd be happy to take any questions you may have.
Yeah, we should say that, yeah, make sure you go to the microphones if you've got a question.
Is it on?
I can hear you.
Okay, so you said it was hard to get increased expression in your cis-regulatory mutants, and so I wonder, is that because you didn't have any repressor sites there that could do that?
Yeah, so I think that's right. I think what we're seeing when we talk about increases and decreases in expression is specific to this gene and how it's regulated. So the cis-regulatory mutations tended to decrease expression, but oddly the trans-acting mutations tended actually to skew towards increasing. So I think what we have is a case where the cis sequences are bound primarily by activators, but in the regulation of those activators there's actually a lot of repression and sort of negative control. And that's something we're teasing apart now.
Hey, Tricia. You posit a variation in fitness with the variation in expression. Have you tried to actually measure variation in the heterogeneity within isogenic lines in fitness directly through single cell stuff or the like?
So track a single cell's doubling time or something like that and equate it to expression level of that single cell?
Yeah, I mean, there are ways to trap yeast. And
yeah, I think you can do it with microscopy. I don't think you can do it with flow cytometry, which is what has been our workhorse. But that would be interesting to do. It reminds me that I've been thinking a lot about if we did track a single cell through time, would we actually see the same distribution of phenotypes over time in a single cell as we see in a whole population? In other words, when we look at a population in a snapshot of time, are we seeing fluctuations that occur over each cell's lifetime? Or are we seeing like some cells take this fate and stay there and others don't? And again, I think we need microscopy to sort that out too.
I have another question. So if noise can be beneficial for fitness because you're sort of at the wrong level of expression, does that also imply that noise could be good in evolution? So if you're sort of evolving and you have sort of more flexibility, and so could that explain, for example, why, I mean, Tata genes, I think, have more noise, but they also change more evolutionary. So is that, you think?
So there is a nice correlation between those. I think Christian Landry's work come to mind for the effects of Tata box and the correlation between those. I think that there are conditions under which higher noise is advantageous for evolution and conditions under which it's not. I think it has to do with environmental stability. I didn't have time to tell you about our work with environment differences today, but we're basically analyzing all those genotypes in 4 different carbon source environments and looking at the differences in fitness curve shape because they do change. And yeah, so I think that again, if you are in a stable environment for a long period of time, it makes sense that the advantageous alleles would be to lock in that phenotype. If you're in an environment that changes generation to generation or year to year, it may be better to maintain some, I don't know if I want to call it plasticity or,
yes,
in that case. Yeah.
Yeah, my question kind of ties to that. So it's been suggested that noise, increasing alleles might be beneficial in a fluctuating environment.
Right.
Fluctuating optima, so to speak. So is that something that could be studied in
we could shift environment, like we could grow for a day in one environment and then shift to another environment and do some experimental evolution. We could do that with this system. We haven't done it. Yeah, good question.
Could this also be related to the statement you made before where transmutations might have one large positive effect but many deleterious effects that are pleiotropic, and that to have the noise in the system that can begin to deal with the pleiotropy negative effects can then fix that positive effect? Have you thought about that at all?
Yeah, so we haven't yet characterized noise in the transregulatory variants in the same level of detail. And the reason for that is the point that I mentioned where our mutants have one mutation that affect expression, but other mutations that when you get into measuring fitness or pleiotropy. So we've now mapped 80 of those mutations, and we've introduced single point mutations, and we're doing, we've reintroduced that, and we're doing that experiment. Yeah.
But I'm thinking that perhaps the cis variation might overcome the pleiotropy from the trans variant. The negative, so that
I see. So that you have a trans-acting variant that maybe takes you to a suboptimal cis-acting phenotype, but you have some noise, and so that can counteract that.
But there's one really positive effect from the trans and a lot of negative pleiotropy. And then the cis could begin to rapidly overcome that negative.
So in some of our studies of cis and trans on sort of genomic level, we see that pattern of compensation. Absolutely. That's actually the strongest signal that we're seeing in some of those evolutionary comparisons in both flies and yeast. So we're trying to sort that out, but yeah. Okay. Thank you.